Yes, recording is okay, so yeah, we have 30 people online, so we can get started, I think. So today it's my great pleasure to have Professor Jen Stowe from Stanford University to give us a talk on his very recent work. So Jen is a faculty member at Biomedical Data Science and Computer Science and Electrical Engineering at Stanford University. He got his PhD in Harvard 2014, and then since then he's been with Microsoft and then later Stanford. So he has won numerous awards, including NSF Career Award, Chan Zuckerberg Investigator Awards, and many others. So he has been working on machine learning and its application in health science and biotech and many others. It's our great pleasure, and especially for this like snowy day, you finally made it. So and also thank you everyone here who are making this today. Okay, thank you. All right, Jen, please. Great, sure. Thanks for the introduction. I'm really excited to meet all of you here. Cool. Yeah, so I want to share with you some of the recent directions we're exploring around how to develop and apply new types of generative AI tools for different scientific applications. Okay. All right. So I hope you know that there's been just a huge amount of really exciting advances, right? Especially in generative AI tools, right? For example, in large language models, now there are many of these different models, and they're seeing quite impressive applications, everything from software, right, to all sorts of commercial applications. But the kinds of applications that I'm the most excited about are usually in these different scientific and biomedical domains, right? So I want to share with you some of the areas we're doing in those domains. And when we're thinking about scientific settings, right, there are usually sort of four types of people, right, that we often work with, interact with, right? So there are the scientists themselves, right? They're often like clinicians or doctors, right? There could be patients, which could also include us. And there are often these different trainees, like students and postdocs. Right, so what I want to do today is to actually share with you some of the work we're doing around how do we actually develop these kind of generative AI tools for each of these four groups of people, right? So for scientists, like for researchers, I want to explore a bit of how we can actually use these tools to really expand the capabilities and creativity, especially for researchers. And I'll give you an example of this for drug discovery. And then for clinicians, we're thinking about how to actually build these kind of visual language models really as co-pilots to help these clinicians to interpret difficult cases, right? For patients, one of the big questions that they're getting a lot of very technical information, so we'll think about how do we use tools like language models to simplify the information for patients. And then lastly, to explore some quite ongoing work around how do we actually use these language models and generative AI tools really as sort of almost like a AI kind of research advisor for students. So there's sort of ongoing experiments that we're doing there. And yeah, so happy for this to be very interactive. So please feel free to ask questions, either in person or online, and just interrupt me at any point. Okay, so diving into the first one, right, around really thinking about how we can use these tools as a way to increase the creativity of scientists. So one area, just to be very concrete, one area where I think it is really important to sort of expand our search space and our creativity is really in the area of drug discovery, right? And just to give you a very concrete example of this, right? If you think about antibiotics, right? The first antibiotics discovered almost 100 years ago by Alexander Fleming, the penicillin, right? And what's really interesting, if you look at the history of these drugs, like antibiotics, is that in the first few decades after penicillin, there's actually a gold rush of actually a lot of new antibiotics being discovered. However, for the last 60 years or so, there are actually very few novel drugs that people were discovered, right, as antibiotics. And as a result of this, right, so now millions of people die every year because they're exposed to drugs, to bugs, pathogens that have developed resistance to the existing drugs that we have. And it's easier and easier not to develop resistance because we don't have a lot of these new drugs. And this is also exemplified by the bottom part here, right? Where in this timeline, right, above the timeline is actually when a new drug's first introduced and put into the market, right? And the bottom part of the timeline shows you when the bacteria develop resistance to that drug. And you can actually see that in many cases, there's only a gap of a few years, or maybe a few decades, right, between the introduction of a drug and resistance. Really highlights the fact that there's this huge arms race, right, between drug developers and pathogens. And unfortunately, right now, we're losing that arms race. So as a consequence, now you have these bacteria like the one shown here above, it's called Acinetobacter baumannii. It doesn't look that impressive, but it's actually an extremely dangerous pathogen because it's actually resistant to many of the existing drugs that we have. It's considered by experts, health experts, to be the perfect predator. So what I want to do here is to say, okay, so how can we think about using generative AI tools to really help us to facilitate and speed up the discovery of new drugs and new molecules? And I want to give you a concrete example of how we actually use this to discover some of the first drugs that are effective at killing this pathogen, that has also been experimentally validated to actually be effective at killing this pathogen, Acinetobacter baumannii. So this work is led by Kyle Swanson, who's a terrific computer science PhD student working with me at Stanford, right? And the idea here is that basically we want to help use AI tools to help us to explore a huge space of potentially new molecules that have never actually been seen before on this planet, right? So if you actually look at the space of small molecules, right, and I'll explain what that space looked like, but it's actually astronomically large. And if I look at all the molecules that have ever actually been seen in nature or on this planet, right, that only covers a very small slice of the space of molecules. So that means that there are actually most of the molecules that we could potentially look at have never actually been observed before. And this is where the generative AI becomes very useful, because we're going to develop these AI tools to help us to efficiently explore the space of new molecules. And then we're going to experimentally validate and test those new molecules that are generated by the AI algorithm as effective antibiotics. So I'll explain in detail how each of the steps work. But before we go in, so the key technical challenge here is that it's actually not too difficult to develop AI tools, right, with diffusion models or other AI models to hallucinate new molecules, right? But the hard part is that we don't know how to make these new molecules, right? If I give a new molecule designed by the AI, my chemist colleagues, most of the time they don't know how to actually create those. And if we can't synthesize these new molecules, then that doesn't really help anyone because I can't really validate these new molecules. Right, so the approach that we're going to take technically is that in addition to having the AI generate new molecules, we're actually going to ask the AI to help to generate recipes for creating each of these new molecules. Right? So you can think of the recipe as basically being like a step-by-step instruction. We're going to ask the AI to teach us how to actually create each of these new molecules. It's not just the final product, but also all the intermediate steps. So just to give you a sense of what we're talking about when we think about recipes for creating new molecules, new drugs. Right, so it's sort of like cooking, right? So you start with raw ingredients and you put these raw ingredients together into more complicated outputs. Right, so the raw ingredient here, basically what we call building blocks or small fragments of molecules, right? So these fragments, you can think of them as being sort of small molecules that are just easily available. You can just order them from different pharma companies. There's over 100,000 of these small molecules, fragments that are easily available. And then there are chemical reactions. So I think of each chemical reaction as basically like a template that can take a subset of these small molecules as inputs, right? And then create more complex molecules, larger molecules as output, right? Again, it's very similar to cooking. You take raw ingredients, you combine them to maybe a more complicated output. And there are orders of hundreds of these potential reactions, where each reaction can work with a subset of the different building blocks. Right? And you can also do these in combinations sequentially. Right? So now you can imagine there's actually a huge, exponentially large space of potential molecules that one could potentially explore. in the dark. By putting together different fragments, different combinations with different reactions. So the space of potential molecules is just astronomically large. And again, in nature, we've only observed only a very small subset of these potential molecules. So what we want to do now is to actually use the AI to help us to explore these new molecules that can help us to design new molecules. So our initial approach for doing this is based on this approach that I'll describe, based extension of multiparallel tree search, right? So some of you might have seen these papers about AI to play games like AlphaGo, right? So behind these AlphaGo algorithms, right, there's actually called multiparallel tree search. You essentially have a huge space of potential moves in chess and Go, and you want to view those as a game tree and have the AI to efficiently explore different parts of that game tree, right? So in our setting, instead of having the game tree being like the space of moves of chess and Go, we actually have the space of molecules, and we want to have the algorithm to explore efficiently the space of the universe of potential molecules. So here's what this tree of molecules will look like, right? So it follows this recipe I described on the previous slide, right? So initially you start with like level one will be building blocks, right? So these are the individual small fragments, right? There's again over 100,000 of these building blocks. And then level two will be take different combinations of building blocks, right? And I can apply some reactions to this, right, to a different combination of building block here, and then create a more complex molecules outputs. Right? So for example, this node here would say take building block one and building block two, and then you apply reaction 27, you get a more complicated molecule as a result. Right. So essentially, right, as you go down from the root of this tree to the leaf, right, you're basically traversing a path, and that path tells you which building blocks to put together with which reaction, right, in which sequence to get more and more complicated molecules. Right. So in other words, each path from the root to the leaf is one of these recipes that we want for creating a more complex molecule. Right. It's the step-by-step instruction. Any questions so far?
Yeah. So are these building blocks, like the molecules and reactions, like real known ground truth ones, so that it is like manually annotated before?
Exactly. Yeah. Yeah. So the question is like, are these building block reactions? Do we know these things? Yeah. So all the building blocks we have here are known building blocks, right? So these are things that we could order them from different companies, and the reactions are also validated reactions. So we know that these things that work in the labs, right? But the space is still extremely large, right? So there's like over 100,000 building blocks with over 100 different reactions, right? And you can do these in different combinations. You have a sort of exponentially large number of structures that you can potentially build, right? And what's difficult here is that nobody actually built most of these structures, right? We only observed a very small number of the leaves that from all the different from nature. Good question. Other questions? Okay. Hopefully the setup of the problem makes sense to people, right? So we want to actually now explore the space of molecules to help us create these new drugs. So the space of molecules here, it's still, as we said, it's very large, right? It's too large to explore. And this is where we need to have AI algorithms to tell us how to efficiently explore this space. Okay. So you can think of this kind of AI algorithm as sort of guiding us to explore this tree of molecules, basically being like a reward model, right? That tells us like which part of this tree is more likely to be promising for generating new molecules. Right? So how do we actually train and build this kind of reward model? Right. So essentially what we do is we call this kind of like a property predictor, but think of this as a reward model. Right. So the way we train this property predictor is by taking all the existing data that we have, right? Where chemists have actually done the screens of molecules against different bacteria. We take all this existing data, and then we train basically a supervised learning model, right, that can try to predict, given a new molecule, how likely is it to actually potentially be a good drug, right? So this classifier is basically a graph neural network that looks at each molecule as a graph, right, and then sort of predicts the drug-like properties of that molecule. Right? And that sort of provides us this proxy for the reward of how good the molecule is. So in this case, right, I go down to this leaf here, and then I apply this reward model and generate some predicted property, right? 0.9 means that it's likely to be a good drug, right? And then that signal is basically updated as the score, right, for each of these intermediate nodes along the entire path. So this is how the information can get propagated, so that if if a fragment is more likely to be promising, right? The model knows that's more likely to be promising if it's more likely to generate a final molecule that has a good score. Now the challenge here is that when we think about building drugs, right? So there are many different competing objectives, right? So there's one objective is like efficacy, right? How likely is this molecule to be an effective drug at killing the pathogens, right? So that's what this property predictor of the reward model is telling us. But in addition, we also want these molecules to be structurally novel, right? So that means that they should be very different from anything that's been seen before, right? And this is important because if it's structurally novel, that makes it harder for bugs to develop resistance to it. Right? It should definitely be safe for humans, right? We don't want these drugs to have toxicity when we take it. And they should also be very easy to synthesize, right? Because if we can't synthesize them, then it doesn't really help. It doesn't help anybody. Right? So you can think of these different objectives now, each corresponding to its own reward different scoring function, right? And all of that is basically combined together into a single score for each node, right? So that's basically how we encourage this model to especially explore the space of the molecule trees.
Yeah, quick question. So for the last property synthesizability, for different blocks, so although they are able to, there's a way to go from block to block, so still the difficulties are different.
Yes.
So okay. So that still needs to be learned. It's not just from block to block, we could directly know that it's the score for it.
That's right. Yeah. So it's a good question. So for example, like this block here is actually blacked out, right? So grayed out, which means that even though it's visible, you can put building blocks 1 and 3 together, but we know that those are unlikely to be compatible from prior chemical knowledge. So that's unlikely to have a high yield reaction. Right, so even though the space, you can literally put any combination of building blocks together, but only certain combinations are likely to have good synthesis rates. Right, so those are actually learned by the model because we have a lot of chemical knowledge we can put into the model to basically prune out different parts of the search space.
Yeah. For those 4 elements, how did you quantify those and where did you get those information?
Yeah. It's a good question. So for basically, you can view each of these as basically its own little classifier, right? So and we train each of these classifiers basically using previous drug screening data. So for efficacy, we can train it based on previous drug screening data that screens different chemicals against bacteria, right, to see which ones are effective, right? And similarly for safety, we can also train another graph neural network based on previous safety data, right? That looks at each of the different molecules. And the FDA has different databases of see which molecules are likely to have side effects in humans.
And then the safety, there's a different, severe, moderate, and mild. Do you also quantify and give the different degree? Since a lot of trials they can fail, but some is really mild safety.
Yeah,
Comparing with others.
Yeah, so it's a quantitative score. So it's so the numerical score that captures the severity or the mildness of each of these. Okay. Okay, cool. So that's how we actually train the model. So hopefully that gives people a sense of what this algorithm look like, right? So, and again, the key takeaway of this, right, is that the output of this model now will actually be a recipe, right? Because it tells us, okay, so this is a new molecule that's promising, and here's the path, right? Or how to go from the root to the leaf of this molecule. That path is basically the step-by-step procedure how to make it in the lab. And it says that the building blocks 1 and 2 together and apply reaction 27, right? So it's a very clear set of instructions. So this is actually what we trained the models all trained at Stanford. So it's a physical model, right? But the fun part, right, is in the pudding. So now we actually get to make these molecules and test do they actually work, right? So to do this, we worked together with a company in Ukraine called Enemy. So they are based in Kiev, and it's very interesting. So this Ukrainian company, they're actually one of the best companies in the world for making these different molecules together, right? And as you can imagine, over the past 2 years, they had a lot of other other concerns beyond making molecules for us, right? But they're actually still active all the time. But because of the work going on, what that means is that everything, the recipes that AI created, that will give to this company has to be extremely robust. And they are not able to change anything or optimize any part of the recipe. They just have to follow the exact steps provided by the AI, and should just have to work out of the box. Right. So we basically sent to enemy 70 recipes corresponding to 70 new molecules that's produced by the AI model. Right. Again, each of these recipes corresponds to an entirely new molecule that has never been seen on the planet before. Right. And then we had this company basically follow the AI's instructions to create each of these molecules. And they were able to actually successfully synthesize 58 of the 70 molecules at a high yield, right? So the hit rate would be around 80%, which is extremely high for these kind of new drug discovery projects. Right, so they actually made 58 of these new molecules, and then they shipped it back to us. And then we had our experimental colleagues, John Stokes, who then validated these molecules. Right, so the way that we experimentally validate them is to basically take each of these new molecules and then test it against a series of different dangerous pathogens to see how effective is each new molecule at killing these pathogens. And then in parallel, we also test these molecules in animal models to make sure that they're safe in animals. Right? There's no toxicity side effects. And the pathogens that we tested, we want to go after the hardest ones. So the World Health Organization had this top tier, the highest priority of pathogens. These are called escape pathogens. These are the most dangerous pathogens because they've developed resistance against existing drugs. This includes this Acinetobacter baumannii, this perfect predator that we showed at the beginning, also includes several other pathogens. So we took 4 of these escape pathogens, and we found that actually there are 6 of these new AI-generated small molecules that are effective at killing all 4 of these escape pathogens. So that's very exciting. And in addition, each of these new molecules, they're relatively safe, at least in animal models. So we put it into animals, they don't show side effects, right? And we observe these animals over time, right? And the pathogens bacteria do not develop resistance to these new molecules, possibly because they're quite novel, structurally very different from existing molecules. But so of course we still need to do human clinical trials. I think this is quite exciting as one of the first examples of entirely new molecules that are designed and synthesized by AI that are then being experimentally validated, right, in animals and also in these experiments.
So does that mean like when training and the model and doing the predictions, the goal is just some more general antibiotic properties, but the outcome is that it can be adapted to fight against the hardest pathogens?
Yeah.
The model is not like optimized or specifically fit for specific pathogens, right?
Yeah, so we did train the model we're trying to steer the models to target drugs that are likely to be effective against these very hard pathogens. I would say that actually the general methodology here, right, of using this AI to generate recipes for new molecules, it's also not limited to antibiotics, right? So we're also applying this to other diseases, like looking at cancer and cardiovascular diseases. Basically, any areas where you think small molecules can be effective, which is actually in most areas of medicine, then we can potentially apply this tool to generate new small molecules.
So what's like the main advantage of this over this sort of novel molecule generation over because I know that there are very large drug databases that already exist with like millions of compounds that you can just order without synthesis. Like, would you, if you like, is there an advantage of this over just applying a property predictor to like some extremely large database already? Do you think you'd get like an enrichment for more effective compounds or something?
That's a good question. So basically how people do drug discovery before was to take existing libraries of molecules, usually on the orders of millions of molecules, and then just screen those. right? And that's where people have mined and found some of the previous antibiotics, right? But basically, we're running out of those low-hanging fruits, right? Because there, the library sizes are still on the orders of millions. But the potential space of molecules that this generative AI explores is just orders of magnitude larger, right? It's hundreds of billions of molecules that would potentially synthesize, right? So that's one thing. And second is that because it's structurally novel, because these are new molecules that have not been seen on the planet before, right? That also means that the bacteria, they haven't had a chance to develop any resistance to these new molecules.
Yeah, it seems like about 20 out of 70 drugs cannot be synthesized. Do you think that, what kind of the reason behind, since my understanding is why you do the synthesize, all those reaction and conditions are based on some current evidence, right? I mean, do you see any why they cannot happen for those 20 synthesized?
Yeah, so there are 12 that
12.
were not able to be synthesized at high yield. Right? So some of that happens just because the experiments was failed, right? Because there are other conditions, like maybe the different temperature and issues with different fragments. There are also just mistakes made by the model, right? So maybe the model actually thought that these certain building blocks are compatible, but in the lab, they're actually not that compatible.
And also curious about the timeline for AI synthesize
Yeah.
to the actual lab and to the.
So I think that's actually the biggest advantage here, because the whole process from training the AI model to actually making the molecules and validating them experimentally took about 4 months.
Oh,
Right, whereas the actual standard drug discovery process would be usually many years, sometimes decades.
and how,
I mean, the algorithm should generate like more than 70, right? So how did you choose the 70 out of the algorithmic generated ones to send to the company to generate?
Yeah, that's a good question. So there really cost is limited by cost, because to actually physically make each of these molecules is expensive. So that's why we only give it 70 to synthesize. So basically, the algorithm generated like thousands of candidates. And then we essentially, and for each candidate, the algorithm also sets a score and a confidence to say like how likely is this candidate to be a good drug. So basically, we just took the top ones. Okay, great questions. So hopefully that gives you a sense of how we are developing these AI tools really to help to expand the search space and create creativity of scientists. So the next part I want to mention is how we actually use these tools as really assistants and co-pilots for doctors, for clinicians. So often the clinicians, like they have a very challenging job because they have to look at all these different new patients, right, with unknown diseases, and have to make diagnosis of what disease does this patient have. And typically, the way that they make diagnosis is often by looking at these images, right? They would, let's say, if someone come in, they'll take a biopsy of this patient, and then generate some of these very rich images of the cells and tissues, and then you have to look at the image and see, okay, does this patient have this kind of cancer or this other kind of tumor? And it's very difficult because these images can come from different patients, different body parts, right? There are many thousands of diseases, right? So it's hard to actually memorize all of these things by the doctors. So we wanted to build like a visual language model, right, that can look at these images and also provide natural language description explanations for doctors to help them make these diagnoses. But the hard part is that, how do we actually get enough good data to actually train and to align these visual language models to make it useful for doctors? So we found interesting a lot of data at kind of an unusual place, right? So it turns out that when doctors encounter images that they're not familiar with, right? So they actually often go on Twitter. So here are some screenshots I took of doctors actually posting images, right? These histology images online on social media, and then asking their colleagues from around the world to help them interpret what's going on in each of these images. Right. So it might be surprising to you that your doctor is actually posting these images online and asking for help. But this is actually recommended practice, right? So first, the images are de-identified, so they feel comfortable putting them online. And also, for example, the US Academy of Pathologists recommend that their colleagues would use social media as a way to share information and share knowledge. Right. And as AI developers, we thought this is also a really terrific and untapped source of interesting high quality data, right? Because here now you have for each image, right, you have this conversation dialogue by multiple experts to interpret this image. Right? And it's exactly the kinds of expert demonstration that we like to use to align to train AI models. And it will be very expensive to get this kind of expert demonstration data. You can get a few hundred, but here are something we have in front of us, potentially millions of high quality expert demonstrations. So the first thing that we did is basically curate this data set. Right? So you can imagine these discussions on Twitter can be very noisy. So the way we curated this is that first we look for specific hashtags that doctors like to use, right? So for example, in different sub areas of medicine, like in kidney or breast or cardiac, right, they have their own hashtags for that sub community. So we curate these different hashtags, right? And then we get basically scrape off data from Twitter from 2006 until recently, right? And so that gives us like a huge pool of millions of potential tweets that are relevant. Right. And then we basically built different strict filterings to filter out low quality information. So we first have like a computer vision model to filter out low quality images. We also have like an NLP model to filter out low quality text. So basically, after we do all this filtering, we still ends up with a couple hundred thousands of Twitter threads that we think are pretty high quality. Right. So each of these Twitter threads will basically have one or more high quality images, along with the corresponding dialogue by multiple experts. Right? So that basically constitutes hundreds of thousands of expert demonstration data that we use to align visual language models. Right, so I think this is also, we made all of this public as well. So this is currently the largest data set, right, where you have these medical images and also high quality conversations and dialogues, often multi-turn dialogues about each of these. So the goal here then is to basically use this kind of demonstration data to align visual language models, right, so that these models can then help the doctors to interpret these images. And the way that we do this, right, is that typically in these visual language models, right, so you have these text encoders and image encoders. And the text encoder here would be basically trained to extract information from the tweets, right, the text of the tweets, and the image encoder is sort of pre-trained, and then we fine-tune it to extract information from these high-quality medical images. And then the key part here is how to align these different models, right? To align the text part with the image part so that they understand what each other is talking about, right? And to do that alignment is where having this paired image and text conversation demonstrations is where it becomes very useful, right? So that's basically we use the data from Twitter to do this kind of alignment. And after that, we basically created this model that we call CLIP, which stands for pathology aligned visual language model. So this is what the example usage of this model look like, right? So this is how our colleagues, doctors at Stanford are using it, right? So they can upload image into the model, and the model in this case would first provide some descriptions about what's going on in the image, right? In this case, it says that there are about 700 cells in the image. There's some inflammation in this image, right? And then the doctor can ask questions, right? And the model will look at the image and do its best to interpret and provide some interpretations. So we also created like a second usage mode for this visual language model, which is more like a retrieval-based usage mode. Right. So in many cases, right, our doctors may not trust the generation provided by the model itself, but they still want to see how their colleagues have discussed similar cases before. Right. So then what we do is basically ask this model to retrieve from all of Twitter, but also from EHR and other databases, right, all of the relevant, most similar cases, right, similar images to the one that the doctor uploaded. It retrieves those images and then summarizes all of the previous text descriptions, right, from these similar cases before, right, and then provides that summarized information, then as information to the user. Right, so even in cases where maybe you might not trust, the clinicians might not trust the model's generation, right, due to hallucination, they still like to see all of these summarized information from the previous cases.
So my understanding for this data set is more kind of classification tasks, right? So you give you a pathology image and then you tell classification. Have you tried this model in the actual pathology image in EHR in the real world data, since there's a corresponding pathology reports. When we compare pathology reports, then we should compare with the social media. There's a huge
Yeah.
difference regarding the description.
Yeah, so it's a good question. So, for example, how some of our colleagues, pathologists at Stanford using this is actually use this to query both the social media and also the pathology reports. So basically look at the image, try to identify the most relevant images. So actually, here's an example, like they will upload the image, it's sort of like a search engine. That's how they're using it. They'll upload this image, and then the search engine will do like semantic search to find all the other cases in the pathology reports in the EHR the most relevant, right, and summarizes the pathology report for the clinician. So you can do both image search or for text to image search.
The second question about the resolution. So in the whole slide image in the pathology is pretty big.
Yeah.
So sometimes we have to do the batch and batch. So in the social media, one of the resolution is that it's the same high resolution as the EHR data.
It's a good question. So it's pretty heterogeneous what people post online. So sometimes they'll post high resolution whole images. Other times they actually just post, they'll crop out like a region of interest and then post those. And so it'll still be a high resolution view, but of just a smaller patch. The other thing I will mention is that the kind of images that doctors post on social media tends to be actually the hardest cases, right? If it's an easy case, then they don't need to ask their colleagues for feedback. So in that sense, the images that are on Twitter are actually the most informative cases for the AI to learn from. It's almost like the human experts have done the active learning to select the most challenging ambiguous cases, right? And then put those online, and then we train on those, right? So the way we actually do it is we first pre-train the model on the more standard textbook cases, and then we fine-tune it on the harder cases that you get from the social media.
Just following on that, do you mean the pre-training on like more general labeled image data that can cover more space? So that it knows general things and then specifically fine-tune for hard cases.
That's right, yeah.
Okay.
So the very initial pre-training was actually done just we take the CLIP backbone, which has been trained on billions of just non-medical, it's general image text pairs.
Oh, okay.
Yeah.
And another question, in last slides, I saw that in the output, there are quantitative description, like 700 cells, 9.9 fold. If it's just like a visual and text pair, how are those generated? Are they like?
Yeah. So the flip model, we view this as a kind of an agent, right? So we give the agent also access to other pathology tools, right? In particular, it has access to another pathology tool that can count the number of cells, and then can also identify the cell types, right? So it's trained on a text image pair, so it knows a lot of the information by itself, but we also give it access to these specialized tools to get these more quantitative metrics.
Yeah, okay, that's cool. Is it like really like calling some APIs or using some tools to calculate, or is like searching in a database like random?
Yeah, so it has access to some specialized tools. For example, one tool that we use is Nuclei.io, which is like another tool that some of my postdocs have built specifically for counting the number of cells and classifying cell types. Right, so then the language model has access to that tool, and it can use it to help its rescreen process.
Yeah, so it's like in the real time, the pipeline is also sending this picture in that IO tool and getting the numbers back and integrating into the output. Thanks.
I have a question. Can I ask?
Yes, please go ahead.
Thank you. So in your previous slide, when you were combining the image encoder and the text encoder part, so for your example, you are considering only one single region of interest for a big chunk of whole slide image. So suppose for some example, if you have a big whole slide image and you are extracting several patches, then how do you generate the image encoder from all those patches to combine with one single text encoder?
Yeah, it's a good question. So, fortunate that actually in a lot of the tweets that people use, the users actually end up cropping out the region of interest, right? So then we know that that patch is the most relevant for the text description. Sometimes they actually have multiple patches, right? And conversation about multiple patches So in that case, then we sort of view this as like a multiple instance learning problem.
Okay. Right. Okay. Thank you. Yeah, thank you.
Okay, so cool. So yeah, so now we talk about how we can build these tools for scientists and for clinicians, right? So third, very important stakeholder are really actually patients, right? A big problem that in our experience, we face a lot of patients is that now there's often overwhelmed by the technical and medical information that they get, right? And we thought that this would actually be a pretty good use case of can we actually use language models to simplify medical information for the patients and make information more accessible. Just to give you an example, right, of the kinds of things that patients get, right? So if you go into a hospital, right, if you have to take some surgery or participate in a clinical study, you have to get these pages of PDF that essentially have to sign, right? These are consent forms, right? And these consent forms are often written at like a postgraduate level, right? So it requires someone with a college degree to really understand what's going on in these consent forms. But most people in the US read at about the middle school level, right? So even for us, it's hard to understand what actually these forms are saying. So to address this problem, right, we want to see can we actually use language models to really take the existing consent forms that you get in the hospitals and dramatically simplify them, right? So that makes it much easier for regular patients to understand what they're looking at. Right. So essentially we take the consent forms, so here's our workflow, we take the consent forms that people are currently using, and we trained GPT-4 to basically take these consent forms and then simplify it into a specific way, right? So that will be accessible at about 8th grade reading level. And then we had like a panel of legal and medical experts to look at both the original documentation and also the GPT simplified documentation, compare them to make sure that first, there's no mistakes made by GPT, and second, it also captures all the essential key components that you want to get across. So here's an example of what this looks like, right? So the details are not very important, but this is from like particular like the surgical documentation. Right, so the top part here is a few excerpts that I just took from the original documentation, right? And there's pages of this. But the main point here is that if you actually just try to read what's going on, right, in each of these paragraphs, even for us, it's not that easy to understand what it's trying to say. Right? There's some fairly verbose and some often technical phrasings. And then this will be basically after we do the language model simplification, just explains in much simpler plain English, like what is the operation, what is the risk and benefit. And we did a bunch of studies that include user studies to show that basically after you do this language model simplification, it takes less time to read these forms, right? This reading level basically quantifies what grade of education someone needs to understand this forms, right? So before you do the simplification, the original forms, you typically require like a college level or post-college level education to understand. After we do the language model simplification, it drops down to about middle school or 8th grade, right? And then this reading ease is like, just means how easy is it for someone to comprehend what they're reading, right? And again, you see a huge increase in the easiness of reading these forms after you do this GPT simplification. So the part that I'm most excited about is that this is actually has been deployed. So since September last year, a few months ago, right? So the largest healthcare system in Rhode Island, Lifespan, and they're one of the hospitals that actually deployed our GPT simplified forms. So basically, now every time a patient goes into surgery in Rhode Island, they have to look at these forms. Instead of reading these more complicated forms, documentations from before, now they're all reading our language model simplified documentations. Right, so this is actually, I think, one of the first examples of an actual product, right, from generative AI that's deployed at large scale in the healthcare system in the US. So we're quite excited because I think this is really this sort of the tip of the iceberg. There are a lot of similar kinds of applications where we can use language models to dramatically simplify medical information for patients to make this whole process much more transparent and accessible.
quick question, would the original form still be provided?
If they want to look at it, yes, they can always look at the original forms.
So is this model generalizable to slightly different contexts? For example, post-surgery education. You know,
Yeah.
it's very long, it's very verbose, and a lot of patients have to readmit it to the hospital because
Yeah.
they didn't follow, right? Because especially like transplant or something like
Yeah.
that. Can this kind of, tool be generalized to that?
Yeah, I think that would be a really good use case of this for surgery. We also been using this for clinical trial protocols. So clinical trial protocols are often very complicated, and we can see that we can also simplify the clinical trial protocols for patients, and also helps with patient enrollment.
So I'm also interested in the training process. You said there is legal and medical review. So I'm wondering how efficient the entire training process is.
Yeah, so we thought it was important to have actually both like doctors and also lawyers, so like malpractice lawyers to review the GPT generated forms to ensure that it actually matches all of their expectations. So that's the part that actually took the longest. But I think that was also necessary in order to convince, for example, these hospitals to really deploy these forms quickly. Sure. They feel much more comfortable because it's been reviewed by lawyers and by doctors.
Thank you.
Okay, great.
So, so after the review, do you use that information to go back to your training process, or it's just, yeah, so.
Yeah, so in this particular case, the review is mostly confirming that the training has been good. Right? But we had some earlier reviews also by other doctors that was used then to train the model. Okay. Okay, great. So the last part I want to more briefly mention is actually how we are trying to think about using these tools as a way to really help trainees, right? Just like students and postdocs. So no, I think one area where these models can be very useful is a way to sort of provide feedback on research, right? And we all know that getting timely and useful feedback on, especially on earlier stages of research projects, is often a big bottleneck, right? So the reviews you get from conferences are very lengthy, and if you want to, especially if you are a junior researcher at under developing countries, it's often hard to get feedback from external experts. So to address this, we actually built a system where, again, we trained GPT-4 to provide constructive feedback on earlier stages of research papers, right? So the pipelines that basically people would upload their paper, right? And then we would basically extract out different parts of the paper, right? It could be your supplement section, your methods, your introduction related works, right? And then we give the language models specific instructions and demonstrations, which are often similar to the instructions that maybe the conference organizers give to their reviewers, right, how to review papers. So give similar kind of instructions to the language model. And then we ask it to basically provide constructive feedback, suggestions to the authors on how to improve the work. And then we want to really validate how useful are the feedback provided by the language model. So we did this. One way we did this is to actually look at the feedback provided by the language model and compare that to the feedback provided by the human expert reviewers. Right, so we've collected two groups of papers. One is a basically set of papers, 3,000 papers published in Nature family journals, right? And for each of those papers, we also have publicly available reviews. And then we can basically look at how much, if a human and an AI reviews the same paper, how much do their comments overlap? So that's basically what's measured by this part here, right? The hit rates like overlap. And what the takeaway message here is that basically the overlap between the feedback provided by our AI system and the human expert reviewer is comparable, right? Similar to the overlap in the feedback provided by two different human reviewers of the same paper. Right. So that means that the AI is often raising similar points that the human experts would often raise when they review these papers. And this is also similar in both like the Nature family journals, which are more interdisciplinary scientific applications. And we see the same pattern when we look at like iCLR conferences, which is one of the main AI conferences.
How are you measuring the overlap between two reviews?
Yeah, Yeah, so basically what we do is that for each feedback, right, we can break it down into a set of bullet points of like what are the concrete and sort of atomic suggestions raised by the AI or by the human. And then we basically compare how much do these bullet points overlap between these two sets.
Yeah, you mentioned that you train like the GPT-4, and I assume that you like use your own data to fine tune GPT-4 for these purposes, right?
We give it basically demonstrations and instructions.
So do you actually do like a gradient fine tuning, or you just do like prompt tuning of the GPT?
Yeah, so this would be basically only the prompts.
I see.
I'll mention maybe in a couple slides of how I actually do what other kinds of optimization of the language models. So that's one way we validated the model, right? And then after we did that, so we actually also released this as a tool where we have basically researchers from over 100 institutions, right? They use this tool, it's all free, so they would submit their paper, right? Usually those would be earlier drafts of their paper, and then this pipeline then will provide the feedback, right? Usually in a couple minutes, and then each of those researchers, if they agree, right, they can participate in and fill out a survey that we also send to them just to see how useful did they find the feedback provided by the AI in improving their paper. And the results I think was quite very encouraging, right? From the survey. So about 57% of the time, right? So the researchers actually found the feedback from our AI system to be helpful or very helpful in improving their work. So that's great. And also about like 40% of the time, they found that the feedback from the AI system to be higher quality than the feedback they will often get from some of the human reviewers. And I just want to also be clear that we're not suggesting that this is a way to replace human reviewers, right? I think it's still really important that as a part of the scientific process, we do need to have human experts to independently and rigorously evaluate each work. But I think this is useful at an earlier stage. Right? Imagine if you have a first draft of your paper, instead of waiting maybe for a few weeks for some collaborators to give feedback on that work, right? You can basically get real-time feedback, right? That can help you to improve your work. That's especially useful if you are from from an under-resourced country where it's especially hard to get expert feedback.
Quick question here. So when you talk about helpful, do you try to distinguish or try to understand how people actually take helpful means? Because the company reviewers, typically people think they're harsh, right? They, for example, one whole of the paper and just criticize that one whole and try to reject the paper, right? So, and sometimes they write very short reviews, like 2 lines, right? We heard some news from iClear. So, did you guys try to follow up and try to understand what other factors make people think that these are helpful?
Yeah, that's a good question. So we basically try to look at what are the type of comments that are more likely to be raised by the AI versus by their human counterpart, human reviewers. Right. So this is like a log ratio. So larger than 0 means it's more likely to be raised by the AI. And less than 0 means it's more likely to be raised by the human. Right, so for example, humans are more likely to talk about like missing citations, right? Especially if it's their own citations that aren't the same, they're more likely to raise that.
Right.
Maybe that's helpful, but maybe not. But the AI is more likely to talk about specific implications of the research, right? Because the model has read like all of the PubMed and all of archive, right? So actually, I think it's able to contextualize this particular work in the field more, right? And then it raises more suggestions around how to better think about the novelty and the contribution of this work in broader literature. It's also, I think the human and AI are equally likely to talk about like algorithmic aspects of the paper, like computational efficiency, like running time and different experiments. Yeah, so the AI is more likely to suggest specific experiments, right? That it wants to suggest to run computations.
I see.
Do you think the user feedback can be explained by kind of the seniority of the researcher, like the students find it
Yeah.
more helpful than the faculty, for example?
It's a good question. So we did, I didn't show it here, but we did actually collect the seniority information and we stratify it. I think we saw pretty similar results across the board, for both for say, master students, PhD students, and also for faculties. So maybe I'll just mention one thing and then we'll wrap up and take more questions. I mean, so basically the thing that we're working on now is to which is natural extension, right? So in addition to providing the feedback on specific papers, actually try to help the researchers to improve their research ideas, right? And this is sort of, when you do this, it's like a language model optimizer, right? We have this language model that proposes some initial ideas, and we have this other language model, it's a critic, right? That's essentially like that reviewer that I showed you that looks at these ideas and provide concrete feedbacks. And this whole system can basically iterate on itself, right? Essentially it's basically doing, you can view this as doing gradient ascent, right, in the space of the ideas. And the way that we use this, then we just provide, as a researcher, a human researcher would provide some seed questions, right? Maybe, I'm interested in trying to make these language models more reliable by doing some sort of fine tuning, right? And then after it does this internal optimization, it's able to convert that seed question into something that's the model believes is much more concrete and more interesting. Right? In this case, it says that here's the concrete research idea that someone could do, right? Use QLoRa particular fine-tuning technique, and says that you should use this particular data set, open the system instruction tuning data sets, and then this is now like an interesting research idea that somebody could write a paper about. And then there's a bunch of these examples, but I think this is a really kind of interesting space that I'm excited to explore is basically use this language model, right? Given the capabilities, it's really a co-pilot to not just do the like coding and writing part, but earlier stage of even the very beginning of how to actually generate good research ideas and design experiments to validate those research ideas. And I think those are the more valuable things that these language models help us to do. Okay, so just to summarize and wrap everything up, right? So hopefully this gives you a sense of how we're thinking about building these different generative AI tools to help the different stakeholders, right? From scientists to clinicians to patients, all the way to students and trainees. And we see how we can expand these, for scientists, how we can use these models to really expand the search space to find new molecules. For clinicians, right, these models can really help as assistants to interpret these challenging images. And for patients, I think it's very useful as a way to simplify information for patients. And for researchers and students, I think it's probably the most useful case would be helping us to come up with better research ideas and better experiments. Right? And I want to really thank all of my students that led these works, as well as our collaborators. So thank you very much.
Thank you. Any additional questions, audience? Yeah, so thanks a lot for the wonderful talk. We have looked at a lot of examples from training models based on text and image data, but I also know that you were previously heavily involved with a lot of quantitative genomic data like single cell or just general gene expression data.
Yeah.
Would you mind sharing some of your insights in how we can embed those kind of data into the foundation model context?
Yeah, it's a good question. I didn't talk about it here, but I'm still doing a lot of work on these more genomic sides. On the genomic side, I think one thing I'm very excited about is basically using language as sort of the natural space to project a lot of the genomics information, right? So we have this sort of this work that we call GenePT, which basically use ChatGPT to capture information about individual genes, right? And then sort of project individual cells onto the space of genes, right? So then it's a natural way to capture the semantic literature knowledge about different genes and different diseases. But I think even for genomics, it's a very interesting, very natural thing to do, so to project the genomic information onto the space of language, right? And then if we could do that, then we can basically leverage the power of all the large language models to summarize and to capture the literature information. Thanks.
So my question is regarding the bias about the data training or large language model in the last one, since we all submit paper, we sometimes review, get a good review, some of that not make sense. I wonder, I mean, training data, for sure, there could be some bias, but I also wonder if this tool could potentially identify some bias to review from the editor standpoint.
Yeah. Yes, I think it's a good question. So for example, for iClear, we actually have both the accepted and rejected papers. So the model was sort of trained and evaluated on both the accepted and rejected papers. But to your point, I think we also have like another estimate that's maybe about 15%. If you look at the most recent like ICML and iClear, the most recent cycle is about 15% of the reviews on ICML and iClear have been substantially generated or modified by AI, right? So that's our current statistical estimate. And I think that number is also going to increase quite quickly. Right? So I think as a community, it's a really interesting question. Like now we're in a space where a substantial number of the reviews are actually already generated or modified by AI. Right? And also we know that the papers are also getting modified by AI, right? So how should we monitor the situation going forward?
Yeah, I have actually a related question. So, I mean, you talk about using AI to help us to polish ideas, but we know that AI is doing by and large doing interpolation, right? That means probably it's going to suggest a lot of A plus B kind of ideas, right? So that means end of the day we are going to face this problem where ideas are generated by AI, papers are written by AI, and then reviews are written by AI. So these are going to kick out researchers, right? End of the day. So how to address this?
Yeah, I think it's interesting. Like, I think these A plus B ideas are already very prevalent, even among
Yeah, I totally agree.
Yeah,
sure.
and then I think
Yes.
I agree. I think AI is especially adept at doing A plus B because it has read all of these papers.
Or plus C, maybe.
Yeah, plus E. But I guess I would say that, for example, the first example I gave about antibiotic, right? That's an example of the AI actually going quite far outside of the human knowledge space, right? So because there we're asking it to explicitly generate new molecules that are very different from anything in its training set, right? From anything that's been synthesized before in nature. And it's able to do that pretty well. So I think I am actually quite optimistic about AI. If it has enough domain knowledge, then it's going to be able to do a fair amount of extrapolation, right? And teach us entirely new things.
Okay.
Yeah, so kind of building off of that point, I guess the goal is to allow these tools to enhance our ability to explore larger state spaces. So I guess why do you think there was that slide with the log fold change and novelty was like the criticism that was least explored by the AI reviewers? So I guess why do you think that was the case, and like how would you mitigate that?
Yeah, I think there is something a little bit more specific, right? So usually when it's often a critique that human reviewers like to give on ICML or iClear papers, this is not so novel, right? Sometimes maybe that's justified, other times that might not be justified, but it is kind of a common fallback criticism that often humans give, right? And basically what that's showing on the AI side is that AI is less likely just to give like a very simple statement about novelty, right? That's the criticism. But it's trying to say more broadly, like, okay, what is the implication of this work in the broader literature, right? How does this fit in with the other piece of the work? Which I think is actually maybe a more fruitful way to look at it.
To just follow up on this too, so you could imagine maybe for iClear, for example, maybe not completely, AI will completely replace human reviewers, but you could imagine that they will train a specialized model that this is something that you want and randomly add one additional reviewer to it. And then, because they provide complementary comments to the.
Yeah.
Yeah.
You can imagine this, right?
Yeah, maybe it's more useful than humans. Yes, that's true.
I think something along those lines that I'm very interested in exploring would be, for example, with the archive. Right? Basically, anytime somebody uploads a paper in the archive, then basically have this AI feedback put into the archive platform, so then they can just get real-time and instantaneous feedback on their archive preprints. And then that would be maybe that would be useful to a lot of the authors. Alright. Okay, let's thank the speaker again. Thank you. Thank you everyone for being here.
Hopefully it's so good. Getting smaller, lighter now.